How do genetics cause certain types of childhood epilepsy? Imagine your brain as a busy city with thousands of tiny electrical signals zipping back and forth. Sometimes, these signals get mixed up, causing the brain to send out sudden, uncontrolled bursts of activity called seizures. Genetics play a big role in some types of childhood epilepsy because they can change how these electrical signals are controlled. One way genetics cause epilepsy is through mutations in genes that make ion channels. Ion channels are like tiny gates in nerve cells that control the flow of important ions like sodium, potassium, and calcium. These ions help neurons communicate properly. When mutations happen in the genes that produce these channels, the gates can become too active or not active enough. This disrupts the balance between excitation and inhibition in the brain, leading to seizures. For example, mutations in the gene called SCN1A affect sodium channels and can cause a severe epilepsy called Dravet syndrome. Mutations in KCNQ2 and KCNQ3 affect potassium channels and are linked to benign neonatal seizures. Calcium channel mutations are associated with childhood absence epilepsy, where neurons become overstimulated, causing brief lapses in awareness. Mutations in genes 4-nicotinic acetylcholine receptors can also lead to specific epilepsy types like nocturnal frontal lobe epilepsy. Some childhood epilepsies are caused by mutations in a single gene that is inherited in patterns called autosomal dominant, autosomal recessive, or X-linked. These are called monogenic epilepsies. For example, Dravet syndrome is caused by SCN1A mutations. Rett syndrome results from mutations in the MEC2 gene on the X chromosome. Angelman syndrome involves deletions or mutations on chromosome 15, and Wolff-Hirschhorn syndrome is caused by a deletion on chromosome 4. These syndromes often include epilepsy, along with developmental delays or intellectual disabilities. Not all epilepsies are caused by a single gene. Many involve multiple genes working together, each adding a small amount of risk. This is called polygenic inheritance. Childhood absence epilepsy and juvenile myoclonic epilepsy are examples where many gene variants contribute to the risk. Environmental factors like head injuries or infections can also influence these types of epilepsy. This complex inheritance pattern explains why epilepsy can run in families without a clear single gene cause. Some epilepsies are linked to abnormalities in the mitochondria, the energy-producing parts of cells, which are inherited from the mother. Mutations in mitochondrial genes can impair energy production in brain cells, making seizures more likely. Others are caused by chromosomal changes, such as deletions or duplications of large pieces of DNA that affect many genes at once. These chromosomal abnormalities are seen in syndromes like Wolff-Hirschhorn or Angelman syndrome. There are also severe childhood epilepsies called epileptic encephalopathies. These include conditions like Lennox-Gastaut syndrome and myoclonic astatic epilepsy. They often have genetic causes that not only trigger seizures but also interfere with brain development and behavior. This means that genetics can influence both the seizures and the overall development of the child. Understanding these genetic factors is important because doctors now often recommend genetic testing for children with early onset or hard-to-treat epilepsy. Knowing the specific genetic mutation can help with diagnosis, predicting how the epilepsy might progress, and choosing the best treatments. Some genetic epilepsies have targeted therapies under research or in use, especially for conditions like Dravet syndrome. Family counseling is also helpful to understand inheritance patterns and the chances of other family members being affected. In summary, genetics influence childhood epilepsy mainly by altering genes that control how brain cells send and receive electrical signals. Whether through mutations in ion channel genes, single gene syndromes, multiple gene interactions, or chromosomal changes, these genetic differences shape the diverse forms of epilepsy seen in children. Always remember, each child's situation is unique, so consulting healthcare professionals is essential for personalized care and treatment.